Thank you. It's really a pleasure to be here, and I spent the day taking a tour of the library, and you guys have such a gem here. It's really quite remarkable. I was very impressed, and I'm so glad that you guys all come out and that there's a forum to have scientists talk to the public about their science. I'm very excited about that. What I would like to do is to give you some background of what we've been studying over the last 40 years, telomeres, the ends of chromosomes. And then I'll take you a little bit fast forward and tell you about some of the new things that we've been discovering just in the last couple of years. So the title is Telomeres, How New Technology Shifts Old Paradigms. So I'll start off by introducing what telomeres are. So telomeres are the ends of chromosomes. And chromosomes are made of DNA. In the cell, you have the cell nucleus, where all the chromosomes are. And within that nucleus, each chromosome has ends of the chromosomes that are the telomeres. And the chromosomes are made of DNA, and they encode specific sequences here, this A, T, G, and C, which specify the code for the molecules that carry out all of the cellular functions. So that's what DNA does, is to encode all of the things that the cell has to carry out. Telomeres are the DNA sequences at the ends of the chromosomes. So again, here in the nucleus are all of the chromosomes. And if we blow one of them up, the telomere here is the sequence at the very end. And all of the genes that encode various proteins that function to carry out cellular function are encoded along the length of this chromosome. Telomeres have to protect chromosome ends, and they also have to maintain the length. And I'll be telling you about that length maintenance in this talk. So human telomeres consist of tandem repeats of a very simple sequence repeated over and over. TTA, GGG. So here it is. TTA, GGG, TTA, GGG, TTA, GGG, TTA, GGG. It doesn't encode for anything. It's not part of the code that makes molecules in the cell. Instead, it plays a functional role at the ends of the chromosomes. So the telomere repeated DNA units are lost at each cell division. When the cell divides, it has to be able to copy each chromosome end, and it turns out that the mechanism by which DNA is copied can't reach to the very end of the chromosome. And so as a consequence, these repeats are lost with each cell division. So these repeats offer a bit of a buffer at the end of the chromosome. So of course, this shortening of the telomeres with each cell division couldn't go on indefinitely, or none of us would be here in this room, because our chromosomes would have all been wiped out. And so what I was studying when I was at UC Berkeley with Liz Blackburn is how is it that telomeres can actually be maintained over time? This was just a curiosity-driven question. We just wondered, if we know that telomeres have to be shortening, how is it that they can actually be maintained? And in doing that research, we were able to identify this enzyme, it's a little machine in the cell, that actually lengthens telomeres. So if you have a telomere with a certain number of repeats, and after a number of rounds of cell division, that shortens, this molecular machine called telomerase can get onto that chromosome end and add more of these simple telomere repeats back onto the ends of chromosomes. So naturally what happens is that there's some shortening of the telomeres and then some lengthening, and some shortening and some lengthening. And the telomeres are maintained in this equilibrium over a period of time. There's an RNA component of telomerase, and there's also a protein component of telomerase that is essential for the enzyme activity. So telomerase maintains the telomeres around a defined length distribution. Because of the shortening and the lengthening and the shortening and the lengthening, if you look in a population of cells at all of the telomeres and you cut right at the junction of where the telomere is, and you want to look at the lengths of each one of these different chromosome ends, we have used something called a southern blot. So this is the telomere length measurement number 1. I'll be telling you about 3 different ways that we measure telomere lengths. This is the first one, the southern blot. So what's shown here is molecules, these telomere molecules, which are separated by their length. So here are some markers that are bits of DNA, the smallest one here, larger ones here. And uniquely, this telomere length distribution, there are some chromosomes that are very short, and that's because it's light here. A lot of chromosomes where it's dark that are medium length, and a few chromosomes that are very long. And that's because of this shortening and lengthening and shortening and lengthening. So if we look at that, what we see is, just as I said, there's a few telomeres that are very short, a lot of telomeres that are medium length, and a few telomeres that are long. So this is how we've looked at telomere length for the last 40 years. Another way that I'll be showing you later in the talk that we represent this length equilibrium is shown here. This is basically just taking this curve here and turning it on its side, where there's some short telomeres, a lot of medium length telomeres, and a few long telomeres. So you'll be seeing this representation of telomere length. Why does telomerase matter? Why am I here telling you about these curiosity-driven experiments that we did back in the 1980s to understand how telomeres are maintained? It turns out that telomerase is required for all cells that must divide many times. So I'll be telling you about 2 stories today. One is that during normal tissue renewal, this represents a cell. Some of the cells in your tissues are stem cells, which have to make more copies of themselves to be able to allow the tissues to grow. Other cells will become specialized for certain areas. But for all of these cell divisions that have to happen, you have to be able to maintain the telomere length with each one of those cell divisions. And telomerase is essential for maintaining that telomere length during this normal tissue growth that occurs. The other area where telomerase is very important is in cancer. So we have these normal tissue-specific stem cells that occur in various tissues, and they differentiate into specific kinds of cells. But if there's a mutation that occurs that causes these cells to divide more times than they normally would, those cells won't be able to continue to divide if they don't have telomerase, if they're limited in their telomere length. So telomerase is essential for the growth of these tumor cells. So on the one hand, there's normal tissue renewal. On the other hand, there's cancer. And both of them have to do with how many times cells can divide. So back in the 1990s, we did some experiments where we looked at the telomere length in blood cells of people and compared it over age. So this is the age of the individuals, and this is the telomere length. This was using southern blots that I showed you previously, a number of individuals. And we saw that the telomere length was shortening with age in these blood cells. And this was a surprise because in other organisms, telomeres tend to be maintained, single-celled organisms. And so why would these telomeres be getting shorter with people with age? And that started our curiosity in looking at this decline in telomere length. We just wondered, well, what would happen when the telomeres get to be really short? And so we decided to do an experiment to test what would happen if telomeres get really short. So we used what's called a knockout mouse. So we took mice and we deleted one of their genes for telomerase. And then we could then breed mice that had no telomerase. And those mice that had no telomerase could not elongate their telomeres. And so the telomeres would get progressively shorter. And so when we did that, we have this small image from mouse, and this is telomerase RNA. We had a mouse where we had deleted one of the components of telomerase. And when we cross that to another mouse that also has one of the alleles of telomerase knocked out, we get out a ratio of the different genotypes. The important one is here. These are mice that have no telomerase. And we call that the first generation, G1, the first generation of mouse that has no telomerase. And so we bred this first generation. Generation 1 gave rise to generation 2. Generation 2 breeding gives rise to generation 3. Generation 4, 5, and 6. And when we measured the telomere length, what we found was that there's progressive telomere shortening with each generation. So the telomeres in these mice, with each generation that we're breeding them, are getting progressively shorter and shorter and shorter. And what we found is that in the later generations, the G4, G5, and G6, there was cell death or cellular senescence that was associated with these short telomeres. I'll show you in a minute what happens to the actual tissues. But what this told us is that since there's the cell death in these late generations, but these mice up here also have no telomerase, but they were fine. What that told us is that it's the short telomeres, not the absence of telomerase, that affects the cell viability. It isn't until the telomeres get to be very short that now we have an effect in this cell death. So the short telomeres in these mice caused loss of tissue renewal capacity. So the normal cells that would renew your tissue could not be renewed. And so in the blood, we saw bone marrow failure. In the intestine, we saw loss of the cell lining. In the skin, decreased wound healing. In the hair, premature graying. And in the testes, cell death that led to infertility. So this was telling us what happens when the telomeres get to be too short. And what we determined is that when the telomeres get too short, in the absence of telomerase, initially there's no effect because there's this buffer zone of these telomeres. But when the telomeres get to be very short, now that signals a damage response in the cells. And that damage response then leads to either cell death or cellular senescence. So it's the short telomeres that directly lead to the cell death that occurs in these different tissues. And while we were doing these experiments, these were curiosity-driven experiments, we just wanted to understand what would happen if telomeres get to be very short. And we found all of these effects in various tissues in these mice. And around the same time, in 1990, there were a number of people who had identified families, human families, that had mutations in telomerase. So here's an example of one of those families. So this is the 3-generation pedigree. So this is the grandmother here and the grandfather and their son, and then that son's son. And this individual came into the clinic at Johns Hopkins with a bone marrow failure syndrome. He didn't have enough cells to be able to maintain the blood cells throughout their cell division. And those individuals that are shown in black were shown by my colleague Mary Armanios to have mutations in telomerase. So they didn't have no telomerase, like our telomerase knockout mouse. They just had insufficient telomerase. And after a number of generations, then they have a number of specific diseases associated with this. The very interesting thing about these families is that this individual came to the clinic in his teens. His father had disease in his 30s, and the grandmother had disease in her 60s. So this was precisely what we had seen in the telomerase knockout mouse, where you get a worse effect of the disease in the later generations. And the remarkable thing is, we had been studying this telomerase knockout mouse before we knew that there was any human disease associated with telomerase. We were just curious about what would happen with short telomeres. And so normally, what would happen is people would find a human disease and then ask the question, what causes that disease, and then make a knockout mouse. But we had already pre-made the knockout mouse 10 years before there was a disease. And so as soon as the disease was identified, it was very clear why there was this worsening of phenotype with later generations, because we had already previously shown that with our telomerase knockout mouse. So what are the diseases that are associated in these families? Mary Armanios is still at Johns Hopkins, and she has 140 families where there are these mutations that cause telomere shortening. And so she can very carefully determine what are the effects in humans of having telomeres that are progressively shortening. And what she finds is that there's bone marrow failure, the same thing we found in the mice. There's a disease called pulmonary fibrosis, which used to be called idiopathic pulmonary fibrosis because we didn't know what caused it. And now we know what causes the pulmonary fibrosis. It's the short telomeres. Immunosenescence, so the failure of the immune system because there are not enough of these immune cells to go and fight infections. Susceptibility to emphysema, liver cirrhosis, and gastrointestinal disease, many of the things that we had identified in the mice. So these are the things then to focus on for this particular age-associated degenerative disease. There are a number of other age-associated degenerative diseases that have other causes. For instance, telomeres are not involved in Alzheimer's and other kinds of neurodegeneration because those aren't cells that divide many times. This is one subset of important age-related degenerative disease. So here's the second method that I'm going to tell you about how people measure telomere length. I'm not going to tell you about all 12 of the ways. I'm just going to tell you about the important ones. So this is called flow fish for flow cytometry and fluorescence in situ hybridization. That's a lot of words, but basically, this determines total telomere repeat content in blood cells. So this is the nucleus of a blood cell, and this is a way that you can look with fluorescent dyes at each one of the telomeres in that blood cell. So these are two blood cells, and we're highlighting the telomeres. And so you can look at the total telomere content in each one of the blood cells. And so that flowfish method, developed by Peter Lansdorp in Vancouver, Canada, has become the method that is used for clinical analysis. So in the clinic, when somebody comes in and the physician thinks this might be a telomere-mediated disease, they use this assay to determine whether or not this individual may have a telomere defect. So here's an example from Mary's work, looking at 300 individuals, and the ages are shown here. So this is cord blood taken from newborns, and then individuals of different ages. And you see that same decline in telomere length in the blood cells that we had seen back in 1990. But this way, we can quantitate what is the normal distribution of telomere length in the human population. So this puts confidence intervals, the 99th percentile for long telomeres, the 1st percentile for short telomeres, and you'll notice that there is a range of normal telomere lengths, just like there's a range of normal heights of people in this room. There's not one height, there's a range of heights. The same thing with telomere length. All of these are normal telomere lengths. But knowing what the normal is, you can then determine whether somebody is clinically below the normal length of telomeres. So that's this flowfish assay. So here's data from Mary's paper. Here are those normal distributions. And in some of those families that she's been studying, the individuals that don't have mutations, non-carriers, are clustered in black here around the 50th percentile, which you would expect. However, the individuals that have mutations fall below this percentile. So you can now determine whether or not somebody is at risk for one of these age-related degenerative diseases using this clinical assay of flow fish. So here are the normal distributions in the population. And Mary's lab has shown that if individuals have very short telomeres when they're very young, they tend to have immunodeficiency, bone marrow failure, liver disease occurs a little bit later, and then pulmonary fibrosis tends to occur in the later years because of also accumulated damage from breathing various hazardous things over time. A very interesting additional aspect that is coming to light just in the last 7 or 10 years is that individuals that have very long telomeres have a familial syndrome of melanoma, cancer of the skin, or glioma, a deadly brain cancer. So on the one hand, shortening of the telomeres leads to degenerative disease because the cells die. On the other hand, continued maintenance of long telomeres will predispose you for cancer because the cells don't die. The cells that should be dying are continuing to divide. So telomere length equilibrium plays a critical role in human disease. So I showed you this equilibrium before, where you have the frequency, some short telomeres, a lot of medium length telomeres, and some long telomeres. So there's a short telomere threshold, and when telomeres get below this threshold, then you have cell death that leads to age-related degenerative disease. On the other hand, there's a long telomere threshold, and if you have telomeres that are much longer than this, then you're predisposed to cancer. So this is the classic Goldilocks experiment. Where do you want to be? You want to be right here in the middle. I have that as one of the questions that I ask my undergraduates. They have to answer. It's like, do you want to have long telomeres or short telomeres after I give this lecture? Most of them say right in the middle. So disease can result from telomeres that are either too long or too short. So that brings us to the question, it's really important to understand how the telomere length distribution is established and maintained. How does this whole thing work? If we want to be able to treat disease, degenerative disease, or cancer, we have to really understand the fundamentals. So we know that all telomeres shorten at the same rate due to this cell division. So long telomeres over here are shortening progressively. And the model that is currently the established model in the field is that short telomeres are preferentially elongated. So if these guys are preferentially going up and these guys aren't going up, then by the battle between the shortening and the lengthening, you'll establish an equilibrium. And there's probably some truth to this model, but what I'm going to tell you is that we need to completely rethink this model. We wanted to know what's this molecular mechanism. That's what my lab had set out to do. And in doing that, we decided it would be really fun to take advantage of new technology that was coming out and develop a new way to measure telomere length. And so we started measuring telomere length rather than by a southern blot or by flow fish. We started measuring telomere length and developed a new method using nanopore sequencing. So this is a way to sequence DNA. Many of you probably heard about the revolution that's happened over the last 20 years in sequencing DNA. This is a new method that specifically gives very long sequences from a single molecule. So it's called Nanopore because this is a membrane, like a cell membrane, and this is a pore, P-O-R-E, where the DNA goes through the pore, and you determine the DNA sequence as it's going through the pore. This method was invented at UCSC by David Deamer and Mark Atkinson at UCSC and Daniel Branton at Harvard. So it's kind of fun to be adapting a method that was developed at your own institution for a new use. We wanted to do this because the fact that you get a single molecule every chromosome is going to go through that pore, and you can look at the DNA sequence as it's going through the pore. So this is the method that we developed. All of the chromosomal DNA, and here I just have a cartoon. Each chromosome is a different color. We have all the chromosomal DNA. And then what we did was to add a unique sequence to the very end, to the telomere. It's a barcode, just like any barcode you would see in the supermarket, where you can say, OK, this is the end of the chromosome. So we added this unique sequence to the end. And then we did what's called whole genome sequencing. We just sequenced everything, all of the DNA in the cell. And because we know the DNA sequence, and it's called a read, that's a sequence of a single molecule, is a very long, up to 30 or 40 kilobases, which is very long. And we have the whole DNA sequence. So we can recognize the barcode, because we put that on, we know what it is. We can recognize the telomere, because we know what the telomere sequence is. And then what's adjacent to the telomere is a chromosome-specific sequence. So each chromosome has a slightly different set of genes that are at the end of the chromosome. So this is unique to the chromosome end. So then we can plot the length distribution, and this is what we call a violin plot. A few short telomeres, a lot of medium-length telomeres, and a few long telomeres. So when we did this, and we did this first with yeast, because it was very simple to do the whole genome sequencing. If we measure all the telomeres, this is what I just showed you here, this distribution, and we knew what the telomere length should be. But when we looked uniquely at chromosome 1L, so in yeast you have the left chromosome and the right chromosome. When we looked at chromosome 1L, 3 what we call biological replicates, 3 independent samples of chromosome 1L, they were all much lower than the average length of all the telomeres. And when we looked at 2L and 3L, they were all much higher than the average of all the telomeres. And then when we looked at all of the chromosome ends in the cell, we were very surprised to see that there were uniquely different telomere lengths. Some of them are much longer than the median, some of them are much shorter. The methods that people have been using before, we were only looking at every single chromosome end in the cell. So we could only ever look at this distribution. So if we know that short telomeres are important for disease and long telomeres are important for cancer, here's short telomeres, here's long telomeres. But what about this long telomere versus this short telomere? It's a very different question. So the models for how this length is regulated now have gotten much more complicated. So this is my way of representing that. Each telomere is regulated around its own unique equilibrium. Each telomere has its own idea about what is long and short. So in this large distribution that we used to look at, we can now see that there are some telomeres that are regulated around a relatively short length, and that the longest telomere here can be shorter than the shortest telomere at this equilibrium. So this gives us a new conundrum to try and understand that we need new models of length regulation to understand how this can occur. So we did this in yeast first because it was relatively straightforward to do whole genome sequencing because the yeast genome is not very big. It then took us another 2 or 3 years to develop an analogous method using nanopore sequencing for human telomeres. We had to add a step where we specifically separated physically the human telomeres because the human genome is so big. But we were able to do that, and we wanted to ask the question, are chromosome end-specific telomere lengths conserved across organisms? So we developed this nanopore sequencing method for human telomeres. And one of the things that we did was to look at 147 different individuals. And this is the data that I showed you previously for the flowfish, which is that clinical assay that is used to examine telomere length. And when we did a very similar kind of experiment, but with nanopore sequencing, we got a very similar curve of the telomere shortening that occurs with age. These are the individuals that are cord blood, so there is a difference that you are born with different telomeres, and then the telomeres shorten with age. And yes, we found chromosome end-specific telomere lengths in humans, but on steroids, even more so than in the yeast that we had looked at. So what's shown here is in blue, all of the telomeres in the cell, and the average is 4.5 kilobases. And then if you look here at chromosome 1, the P arm in humans, it's called P and Q. Mind your P's and Q's, I guess. It's petite and Q. Those are the two chromosome arms. So this is chromosome 1, the short chromosome, this is chromosome 1, the P chromosome arm, the version that you inherit from your mother. This is the version that you inherit from your father. So the average telomere length is 4.5 kilobases, and there's a 6 kilobase difference in these two chromosome arms. We had no idea. This was like very, very surprising. And it's not always true that it's the paternal copy that is longer. Here's an example where it's the paternal copy that's shorter. And some of the examples where these two red chromosomes, 3P, have the same telomere length. So this was, again, very surprising and asks the question, how do we think about what a short telomere is that causes disease? How can we incorporate this into our model of short telomeres causing disease? So each telomere is regulated around its own unique length. And now we are just setting out to explore what this means for telomere length regulation in human disease. So here are some of the implications of what I've been telling you. Telomere length is regulated as a distribution. Short telomeres contribute to age-related degenerative disease, while long telomeres predispose to cancer. Previous methods to measure telomere length could not distinguish between chromosome ends. Nanopore sequencing allowed us to detect end-specific telomere lengths. New mechanisms for telomere length regulation can now be explored. So this is a theme here that new technologies are transformational in moving past old paradigms. If we didn't have this new way of looking at telomeres where we could look at each individual chromosome end, we wouldn't know that there are these vast differences in telomere lengths. So it's a new horizon for us. We've been working in this field for 40 years, and I could not have imagined that this is where we would land. So I want to show the very important, the people in the lab that contribute to all of this. And my lab has been funded by the NIH for the last 35 years. And that NIH funding actually supports the training of these scientists. So these are my current people in the lab. And about 90% of the money that I get from the NIH is going to training this next generation of scientists that can go on and understand how it is that short telomeres may cause disease. So I've had continuous funding over the last 35 years. Since 1989, I got my first grant when I was 27. And I had that grant, and then I got a grant from the National Institute of Aging. I had that for 26 years. Another grant from the National Institute of Aging for 5 years. I then branched out into cancer and tried to understand how telomeres play a role in cancer. I had that grant for 5 years, another cancer grant for 7 years, and my current grant from general medicine to understand these fundamental mechanisms underlying chromosome-specific telomere lengths, started a couple of years ago and will end in 2 years, and that might be my last NIH grant because the NIH isn't funding science right now. Most of what I do is training the next generation of scientists. 90% of that grant funding goes to support the students and the postdocs and the specialists who are doing the science. So here's some examples of other lab photos that we've taken over the years, and a few additional examples of the scientists and their babies who have trained in the lab, and yeah, we have fun too and go tubing on the river. So training the next generation of scientists. So I have on my website this timeline of all of the scientists that I've trained over the last 35 years. So Lee Harrington, she was my first graduate student, is now the chairman of the department at the University of Toronto. Let's see, Kathy Collins is a professor at UC Berkeley. Michael Heeman is a distinguished professor at MIT. The list goes on. Other individuals I've changed. David Felser is a professor at University of Pennsylvania. Chris Frank is a professor at the University of Minnesota. So a number of people over the years. These are the scientists that are going to be making the next discoveries that are the most important discoveries to bring us forward to understanding health, disease, and other things that we can't even imagine. Right now, funding for science is under threat. Trump administration blocks NIH from awarding any research grants. This was a few months ago. NIH has 2 months left to spend. How to wreck the nation's health by the numbers. NIH is shrinking the number of research projects it funds due to the new Trump policies, and executive orders seeks to centralize control of grant making. This is probably why my research funding will end after 40 years, coming 2 years when my grant is up. The war on cancer was highly successful. Many new drugs, many targeted therapies for cancers came out over the last 35 years. And now, the National Cancer Institute will fund 4% of submitted grants instead of the usual 15 to 20. This has an implication for healthcare. If we're not doing the research, we can't cure diseases. So here's an example of what the planned cuts to these different institutions. So heart disease, respiratory disease, diabetes, cancer, there's a 37% cut. Stroke, a 40% cut. So these all will have to do with the health care that we will have going forward over the next many years. Here was a particularly striking number that I found in terms of the life expectancy in years from 1980 to 1922. So this is the United States here in green, and this is comparable other countries. So a lot of European countries and Canada. There was a big drop during COVID, but we're still quite far from other countries. So there's still a lot that we need to be able to do to understand and treat disease to be able to maximize our health span. So if you can, and you have an opportunity to talk to anybody that's involved in these things, please support the national funding of medical research like your health depends on it. Thank you.
All right, everyone brace your eyes. The lights are about to come up. If you have a question for Dr. Greider, raise your hand and give me a moment so the virtual audience can hear you as well.
Fascinating talk. I mean, I'm so glad I came and heard this. My curiosity is obviously a lot going on now has to do with AI and how that's supposed to be increasing the ability to take lots of data very quickly and quantum computing. How do you see that affecting what you're doing?
It is the best of times and it is the worst of times. AI has already been transformational in molecular biology and biochemistry. One of the earliest applications of AI had to do with predicting protein structure. There's a program called AlphaFold. Last year, they got the Nobel Prize because you can just take that DNA sequence and determine what the three-dimensional structure of a protein is. I never thought that would ever happen in my lifetime. We were told you could never do that when I was growing up. And by doing that, you know what the three-dimensional structure is. You can then find drugs to target proteins because you know their structure. So you can find a structure that will fit in and inhibit a particular protein. It's been transformational. So that was about 5 years ago. Those things are still going on. We started collaborating with a group at UC Santa Cruz who is using AI to predict sequences. We want to ask the question, are there sequences just next to the telomere that might influence the telomere length of that chromosome? I don't know a lot about AI, but it's very powerful, and so we're doing a collaboration to see if we can use that. So on the one hand, that's very helpful. On the other hand, for my teaching, my undergraduates and my graduate students, it's very disruptive. So, but it's here to stay, and it's going to be transformational. In the medical field as well, people are using AI to look at radiology images and other things. There will be changes happening faster than we can ever imagine changes are going to happen.
Thank you. On the, you kind of stole my first one on AI, so that covers that. And in the area of, you had mentioned 12 diseases, or if that many, macular degeneration in the eyes, is that one that's significant that you will be able to do research on, or your trainees will do research on in the future, and hopefully the near future?
There are a lot of people studying macular degeneration. That's not one of the things that is caused by the telomere shortening. The nice thing is that well, we have the advantage that we have these families that have mutations in telomerase. So all we need to do is study those families very carefully to know what are the diseases that are going to be caused by short telomeres, because they have short telomeres. So we know the constellation of the specific age-related degenerative disease that is caused by the short telomeres. And those individuals do not get macular degeneration. So there's a different mechanism. There are several different mechanisms that are going to be involved in age-related degenerative disease.
Right in the middle.
Yeah, thank you. Is there something special about the end of the telomeres that, like when they're shortened, do they lose a whole, that sequence you described at the beginning, or does it kind of end somewhere in the middle?
So a human telomere is, say, 6 kb long, and it loses maybe 100 base pairs every cell division. But then telomerase, in cells that have telomerase, can just top it back up. So there's a shortening and lengthening. We're just really trying to get at the question of, what is the short telomere that triggers that cell death? We don't have the answer yet. In yeast, it looks like it might be around 75 base pairs. In humans, maybe a little bit longer. It could be that different organisms have different triggers for how short is short. But that's a question that we're really digging into the data that we have right now to answer.
Thank you very much for an interesting discussion. David Weinberg, I believe is the name, wrote a book, Biology of Cancer, that I've been reading, and it's got a chapter on telomeres.
Bob Weinberg, yes.
Bob Weinberg.
Yes, exactly.
Okay. So my question is, he was commenting that it's primarily the short telomeres that are causing cancer. On your data, you actually have long. So what additional mechanisms are you aware of that he's not aware of in his text that are causing cancer in individuals?
Yeah, there was a lot of experiments where people thought that the short telomeres might then cause chromosomal rearrangements in the cells that could lead to cancer. And all of those experiments were done in what we call tissue culture cells, so cells in culture where they would induce short telomeres. When you now look, and my colleague Mary Armanios, who has all these patients, when you look in patients with short telomeres, they do not have elevated cancers. So it was something that, in culture, the experiments that were done were valid. They interpreted those, but that doesn't have to do with the whole organism. So in humans, it's the long telomeres that cause cancer. The short telomeres do not cause cancer. The long telomeres, it's all about cell division, right? So the short telomeres shorten and you get cell death and age-related degenerative disease. The long telomeres, you do not get cell death. The tumor cell, you get a mutation, the tumor cell starts growing, and now it has enough telomere left to grow for a much longer time. It's all about the number of cell divisions.
All right, back here near the Taza.
Are there any medications or drugs being developed to cure this or potentially slow down it?
Yeah, so the question is, are there drugs? And the answer is, people have been looking for a long time. When we started understanding that telomeres play a role in cancer, there were a number of studies trying to find drugs to target telomerase to inhibit cancer. And that has not panned out to any drug that is actually targeting telomerase. Conversely, you would now want to be able to elongate telomeres to treat the age-related degenerative disease, but not too much, because then you'll get cancer. But the answer is no, there aren't currently any drugs out there. And what we're thinking is that we really have to understand the fundamentals here. How is it, like we didn't even know that each chromosome end had a different length. We have to understand what that mechanism is, and that will give us a handle then to be able to capitalize on that mechanism and develop new drugs.
And we'll stay at the Taza.
That's such an interesting discussion. So, the telomere length equilibrium, that is so important. And you mentioned about the mutation of the telomeres. And I'm wondering, I think you probably have the information. There are several kind of mutations in telomeres, which would be like, and resulting into insufficient telomerase or gain of function of telomeres or loss of function of telomeres. And how, I just wanted to know that if these are all the different kinds of mutations in telomeres is important for the telomere length, equilibrium.
Yeah, what we find is that the same thing that we found when we did our telomerase knockout mouse, that what matters is the telomere length. There are a number of different genes that contribute. It can be telomerase, it can be a telomere binding protein, but it's how short the telomere is that determines what the disease is, just like in the mice. The early generation telomerase knockout mice didn't have any disease because their telomeres were still long enough. And you have to get the telomeres to be short, and then you see disease. So it's not about a mapping of a particular mutation to a particular disease. Rather, it's the short telomere to the disease.
Back on your right.
Yeah, thank you for a really nice talk. I'm a graduate student from KU Medical Center, so I really appreciate your effort in training us new generation researcher. Yeah, and I'm really sorry for NIH funding.
Hang in there. Hang in there.
I'm really sorry for the NIH budget cut. I think they should recover it. Yeah, I work on neuroscience, so I have a question. So for post-mitotic cell, like neurons, they do not enter the cell cycle. Therefore, their telomere lengths are relatively stable. Yep. However, some studies show that their telomere can still change. They are not the same length due to the stress-induced DNA damage or structure instability. But it's hard to measure. With the next generation sequencing like Nanopore, do you think for post-mitotic cell, we can use it to show the stress level because it can accurately measure the telomere length?
I definitely think that using Nanopore will allow us to look at many different tissues, including neuronal tissues. Any kind of tissue that you can get high molecular weight DNA from, we should be able to do this. We're just starting with some collaborators to look at other tissues. In the past, it's always been blood cells because we had this flowfish assay, which was using white blood cells. And now I'm really hoping to promote this method to many other people so they can start measuring telomere length in a lot of different settings like you're talking about.
We'll stay on your right.
Hello. A long time ago, I listened to a talk where someone was talking about the mechanism of senescence of egg cells.
Senescence of
egg cells in the ovary, and because they age, they're stable for a long time and then fairly quickly age. So, is there an association of telomerase length with aging of ova?
There's not evidence that the telomere length plays a role in that setting because the cells aren't dividing, right? These cells are arrested in meiosis, the very last step. And it's thought that perhaps degradation of how the chromosomes are segregating will play a role there. But it's only in cells that are actively dividing that you would see the telomere shortening.
Stay on the right.
Thank you for your passion. In a family where the mother died in the late 70s, the son died at 60, and then his son in his 30s is showing signs of degenerative disease. The other two died of pulmonary fibrosis. Just wondering, are there tests that these family members should be taking, or are you still working on that for prevention?
So my colleague Mary Amanios, who did all of the clinical studies, is still studying these families, and there is a constellation of specific diseases like the pulmonary fibrosis, the immunodeficiency, and if you see a collection of those in a family, that would send off the alert. And there's now, at Johns Hopkins, they have this clinical test, the flowfish test, to determine, is this somebody who actually has the short telomeres? So whether or not the Nanopore will make it into the clinical things, that's not something that I have the capacity to do without having a clinical setting, but there are people that would like to do that. But currently, anybody, any physician that thinks that this constellation of diseases in a family might be short telomeres can get the assay, within a week.
All the way back past the Taza.
Hi, thank you for that talk. I was wondering if you could talk about the work by, I think, Geronimus and some others that look at chronic stress and its impact on telomere length, and could talk about what's the exact mechanism for how that works.
Yeah, so that's one of the reasons I've been talking about these methods for measuring telomere length. There's a very popular method that's called quantitative PCR that people have used for a lot of the studies that have looked at exercise-induced telomere length changes and stress-induced telomere length changes. But most of those changes are within that range of normal that I was showing you. So they're measuring changes, but the assay also has a lot of variability. And so I think it remains to be determined whether or not there actually are meaningful changes in those settings. So now having a very robust assay where you can just do DNA sequencing should be able to bring clarity to a lot of those claims about lifestyle changes that might affect telomere length. So I'm looking forward to that.
Towards the front over here.
Hi, I was wondering your thoughts on treatments where they boost partially function of telomerase, or if you think that it's too risky with the predisposition to cancer.
I think that there is room for both inhibition of telomerase in certain cancers and for possible elongation of telomeres in some degenerative diseases. Now that we have a way to stratify patients, and if you knew somebody was down below the first percentile for normal telomere length, you wouldn't want to give them telomerase inhibitors because they may then suffer some of the consequences. But if they were, above the 50th percentile or higher, so by doing what we call precision medicine or, individualized medicine, and knowing what the telomere length is to begin with, I think that there's definitely room for developing treatments on both sides, the long telomeres and the short telomeres, just doing it very thoughtfully.
Thank you so much.
All right, we're going to stick around the same area.
On the risk of getting into metaphors, what does the telomere do? And I kind of think of it as the end of a shoestring, like it keeps the shoestring intact, or is it during mitosis the hook that the process used to pull the chromosomes apart? Do you want to risk?
Well, the shoestring analogy, the end of the shoestring is called an aglet, and that was something that was developed when I was at Cold Spring Harbor, and the science writer at Cold Spring Harbor coined that term, so it's been following me around. But we know that the telomere gets shorter and shorter and shorter and shorter, and then the damage occurs. So it's more like that shoestring analogy in that the short telomere, we know, triggers what's called a DNA damage response, and that then leads to the cell death or cellular senescence. And so, with the shoestring analogy, the whole thing falls apart when it gets short. So it's similar to that.
Okay, thank you.
All right, we're coming up on the hour, so we'll do a couple more.
how do we keep the telomere length just right, neither too long nor too short? Do you have suggestions?
I would love to know the answer to that question. I think that by now, by focusing on, what is the mechanism that changes? How come one chromosome has a very different equilibrium and another equilibrium? We might find a molecule that is responsible for that. And so then we could have ways of manipulating it. That's what I'm writing my current grants on.
Right, up at the front.
There's many cells in the body that are programmed for cell death, and they call that apoptosis. Is that any relationship to telomere length at all?
No, there's a whole process of specific cells, like when you're a baby and developing, that are undergoing programmed cell death. This is also called apoptosis, but when there's too much DNA damage in the cell, rather than continue with DNA damage, because you might then go on and have other incorrect things and contribute to cancer, the cell engages that pathway, but it's due to the DNA damage. And the telomere looks like DNA damage when it's short. And so it's a specific pathway, but it's a DNA damage-induced pathway, not an embryological determined pathway.
Okay, we'll stick in the front here.
Yes, very, very quickly. A moment ago you mentioned lifestyle changes, and I'm wondering about impact, and I'll use exercise and then my favorite, sleep. Don't both of them have a benefit for some of the things you're talking about?
There are a lot of papers that suggest that there are things that you can do to change your telomere length. But again, I showed you that distribution of telomere lengths in the population. All of those changes are within the normal distribution. So we don't yet have any evidence that there's a change that's a meaningful biological change that occurs. I'm not saying that there's not. I'm just saying that there isn't yet any evidence for it. And now that we have a much more accurate way to measure telomere length, I think we need to go back and revisit some of those questions. But it's still good to sleep and get exercise and meditate and eat well. All those things are still good. I can definitely say that.
All right, up at the front.
Dr. Greider, first of all, thank you for bringing up the limitations on research. I can tell you that us area universities are feeling the stress in a big way.
Yeah.
The question I have is, why does the length decrease with age? Does the telomerase get lazy with age?
So it's really interesting. In the white blood cells that we originally saw, back in 1994, that when you look at older individuals, the telomeres are shorter. You have just enough telomerase in the cell to be able to maintain telomeres for most cell divisions. But in the blood, the number of cell divisions is just a few more cell divisions than the amount of telomerase. So in some of these patients that have these telomere diseases, they have 10% less telomerase than a normal person. But over many generations, that accumulates. So the equilibrium, the Goldilocks, is so finely tuned that that's a particular cell type. Most times, it doesn't matter, because we all, you can get to be 90, 100 years old, and most people won't ever run out of the telomeres. But the individuals with the short telomeres will in the blood. So it hasn't been selected against in evolution, because it would take many, many, many generations, right? There's some very, very devastating diseases that aren't selected against in evolution. This would take 5 generations for you to even see the evolutionary consequence. So the cells have telomerase, but just not quite enough.
All right, let's make this our last question. From Beth online, she asks, where do you think the next major discovery in telomere research will come from? Genetics, epigenetics, or computer modeling?
All of the above. We are doing all of those things.
Excellent. Come around here. Thank you so much, Dr. Greider.
Thank you.
And thank you all so much for joining us tonight. For all information about our upcoming programs and events, visit lindahall.org. Enjoy the rest of your night.